Hello, hello. Welcome everybody to my very first YouTube video. This is completely authentic. I only have the bulleted points in my head, so I don't know if that counts as making it a script. But what what I'm doing right now is filming my study journey. So, context: I am currently a Sec 4 senior in my school, and I'll be studying for my O-Levels. It's coming in less than a month. So I'd like to dedicate this whole month to purely study, like absorbing all the content and blurting them out, and being able to blurt them out during my my exams. So I will just share the subjects that I'll be sitting for. The very first one will be biology practicals and chemistry practicals first, then biology practicals. So yeah, pure chem and pure bio are my subjects. I have paper one, two, and three. Three being the practical, and one and two being the MCQ and the uh paper two is the word. Is it called word problems? Problem solve word problem like yeah, whatever. Paper two is the paper two. Then after other than that, I also have my English. So I have paper one, two, and three. Paper four, I've already completed it. O levels, oral, and my Chinese I already finished the whole thing, the whole shebang, and I managed to score like a C5 merit. Oh my goodness! And I've gotten a oh yeah, I've also got my humanities, so that's my elect history and elect jog, elect history, elect history and elect social studies. So this is going to be a wild one. Because to be honest, what I did for my prelims, I already finished my prelims by the way. What I did for my prelims was that the night before I was like chonging everything, and it's hectic. I don't want that to happen on the day itself. So that's why I'm making this video lah. So hopefully, because I'm uploading them and I'm recording these videos, that I would like find this as a sort of self motivation. So yeah, I really do think that you guys can help me out on this, and hopefully, if you see me constantly posting, that means things are going well. I hope. Okay, so how it goes is that I will share my objectives for the day first, my objectives before studying, and then after that, I will share with you what I've learned um, through this study session. So right now, I'm actually studying biology. I'm studying enzymes, and I'm actually halfway through studying enzymes, and I believe that give me like I don't know, an hour more, and I'm able to complete the whole chapter. And after that, I'll just share with you whatever I've learned, and basically kind of like do like a blurting session. Okay, so sorry, my siblings just came in, and yeah, I think that's about it. No, what happens is that I'll study one chapter. I'll study one chapter for like a specific subject. I'll blurt it out. I'll say it to the camera, and then I will study one chapter of a different subject, so that at least it kind of balances out. Because it's a bit of a waste if you were to um, only prioritize one subject when you have also a lot of other subjects. It's going to be very I don't think it's going to be a very smart idea to just prioritize one subject, because honestly, my attention span for just one subject is crazy little. Like this small, like this much. I also got this mic. Oh my goodness, yes, costed me $10 in Challenger. Oh my days, and yeah, I don't know. I have nothing else to say. All right, okay, I'm back, and okay, I'm gonna just put it there. The thing I'm using for revision is this, the Biology Matters Revision Guide. What edition is this? Mm, oh wait, this is based on 2023 syllables. So I know this year is 2024, but 2025. But my friend got me this last year during my birthday. Guess what? I'm 16 now. Oh, what's going on? He got me on my 15th birthday, by the way. So I'm gonna close this book because I've got in my head. Trust me on this. All right. So enzymes. The very first component is talking all about what exactly is enzyme. What do they do? So an enzyme is basically a biological catalyst, and a catalyst, um, or rather, the catalyst aspect is that they speed up, or rather, they change the rate of reaction. They alter the rate of reaction without itself being okay. Hold on. Without itself being changed after the reaction. So uh, that's actually the definition of a catalyst, which is that they speed up the rate of reaction without being while remaining chemically unchanged at the end of the reaction. Um, I'd like to point out that there's a reason why we usually say change the biological enzymes. They change the rate of reaction, not speed up the rate of reaction. It's because that um, there are times when enzyme catalyst or rather the uh, catalytic effect of enzymes is actually to slow down the rate of okay they're called inhibitors if I'm not wrong, and an example of this is like yeah, so I'm so sorry. Just put it out there. Inhibitors exist, and because of that, we want to stay safe and say that they change the rate of reaction, not speed up the rate of reactions. Because there are times when inhibitors, in the presence of inhibitors, right, catalyst actually slows down the rate of reactions. They inhibit the rate of reaction. Yeah. So put it out there. Yeah, that's one thing. The what makes it biological catalyst is that they are actually found in living things, and they are made out of proteins. So yeah, this goes for all enzymes. They are found in living things, and they are made of proteins. Now, I'm supposed to talk about the activation energy. Okay, so enzymes they speed up or rather they change the rate of reaction by lowering the activation energy. Um, what is an activation energy? Well, the activation energy is actually the energy required to uh, to start the reaction. So in an energy profile diagram, right, it goes something like reactants, and then we have a higher activation energy, and then we have the products. So the higher activation energy is without the presence of catalyst, whereas in the presence of catalyst, right, the activation energy is actually lowered, so it goes lower, smaller bump, and it goes down. So what this entails is that because or rather in the presence of catalyst, right, the the presence of an enzyme, the activation energy required for the uh, reaction to start is lowered, and my, my mom just went by. Whew. And what else? Yeah, okay, so yeah, that's how the enzyme speeds up the rate of reaction by lowering the activation energy. And oh dear, okay, I have something I want to say. I've got to remember. Hold on. Mm, this is my remembering phase. Hold on. Okay, so activation energy, activation energy, activation energy, activation energy. Oh yeah, okay. So uh, hence because they lower the activation energy, right? The rate at which the products are formed actually increases. So it takes a short amount of time for products to be formed in the presence of a catalyst. And uh, in okay, so the so there are two types of components in an energy like in a enzyme catalyzed reactions. And the first component the first group of enzyme catalyzed reactions is actually anabolic reactions. So actually there are two, anabolic and catabolic. Oh my goodness, I should structure it properly. I'm so sorry. So yeah. I don't know, this is all just coming from my brain. Okay, so anabolic reactions, they're basically um, reactions which involves the or rather anabolic reactions are um, basically when it's basically a process which converts um, simpler molecules, simpler substances into a larger, more complex substance. Okay, in the text, in the revision guide, they say like something about a larger, complex molecule synthesized from simpler ones. I'm just putting my own words, and it makes it so confusing. Oh my goodness, but it helps you understand, I guess. I don't know, man. Okay. Now, for catabolic reactions, it's kind of the opposite. It is when you get the larger, more complex molecules and substances, and you break it down into simpler substances, lah, essentially. So the next component is examples of anabolic and catabolic reactions. So for an example of anabolic reactions that I like to give is when special enzymes in the cytoplasm of a cell, they can actually synthesize proteins from amino acids. So amino acids, polypeptides, proteins. Yeah, they're present in the cytoplasm of cells, and yeah, that's an example of anabolic reaction. There's another one. Um, it's when enzymes. There are actually some enzymes that are responsible for the synthesis of glucose from glucose, yes, synthesis of glucose from water and carbon dioxide during photosynthesis. So there are two types of examples that I gave us, and that is synthesizing of proteins. Second one is synthesizing of glucose for during photosynthesis. Yes. So the second component is catabolic reactions. Now, catabolic reactions, we've got quite a lot to say. So the very first example of this, if I look here, I'm looking at you. But then if I look at my face, I'm like, okay, no, this is me looking at the camera, like camera itself, and this is me looking at my face in the screen, like oh, okay, I should stop. I'm using this as an excuse to think, to remember. So, catabolic reactions, okay? There are three examples that they gave us. The very first example is okay. The first one that I can think off the top of my head is actually the detoxification. But okay, let's go in sequence. Okay, so the very first thing that they said was actually the enzymes known as digestive enzymes. Some enzymes known as digestive enzymes actually breaks down larger food molecules into simpler ones, smaller ones that are soluble in water and can actually pass through the cell membrane. And yeah, it's important for absorption, right? Um, Absorption through the villi into the blood capillaries and lacteals. Okay, wait. Sorry, I ate like a walnut cookie just now, like two of them. It's really good. Okay, so the second example of our catabolic reaction is is the detoxification. Okay, so it's an example of detoxification. So uh, larger toxic substances are broken down into simpler, safer ones by like some specific enzyme, special enzymes. And an example of this is the byproduct hydrogen peroxide that can be produced in during certain reactions in the plant and animal cells. And this hydrogen peroxide, it's toxic to our if it's present in the cell. So what this enzyme does is that um, they actually break down the hydrogen peroxide into oxygen and water, which are neutral, but right, yeah, and they're not toxic to us, and they're safe for us, and they're smaller. So catabolic reactions, yeah, fits the description of catabolic reactions. And these oh, the enzymes are called enzymes responsible for this catalytic conversion of enzymes responsible for the catalytic conversion, catalytic breakdown of hydrogen peroxide into water and oxygen is actually called catalase. And catalase is particularly abundant in the liver and blood of mammals. Yeah. Whew. Now, lastly, is that there are some enzymes which are responsible for the breakdown of glucose into carbon dioxide and water. Carbon dioxide and water. So it's like the opposite of photosynthesis, and that's cellular respiration. Another example of catabolic reactions. Okay, so we are done with both anabolic and catabolic that I can remember, and now we're moving on to the digestive enzymes. So I don't know why, but in the book they gave us examples of carbohydrates, protease, and lipases, and they are basically examples of digestive enzymes, which are responsible for carrying out catabolic reactions. Right? So I think they're just trying to elaborate on that, and I I memorized them. Yes. So for the function of carbohydrates, it is to break down the it is to digest carbohydrates. For proteases, is to digest proteins. For lipases, is to digest lipids or fats. And okay, so for carbohydrates, right, there are a couple of different specific types of enzymes. The very first one is amylase. Yeah, amylase is responsible for breaking down starch into maltose, and the maltase is responsible for um, the digestion of maltose into glucose, and sucrase is responsible for the digestion of sucrase into fructose and glucose. And lastly, lactose is responsible for the digestion of lactase. Sorry, lactase is responsible for the digestion of lactose into glucose and galactose. So yeah, so if you are lactose intolerant, well, that's because you lack lactase, and because of that, the undigested lactose actually travels up the colon and gets fermented by bacteria, which can produce gas, acids, as well as the main cause for your diarrhea. So yeah, continue drinking your milk. Okay. Um, um, now we move on to proteases. Protease. Okay, so there are three types that I could think of. The first one is pepsin, and it actually pepsin and trypsin actually kind of like their responsibility is kind of like the same thing. They actually they both convert or rather digest proteins into polypeptides. So the main difference between pepsin and trypsin is that pepsin is their optimum pH and their inactive form, I guess. So for pepsin, their inactive form is pepsinogen. For trypsin, inactive form is trypsinogen, I think. Okay, and for pepsin, it is actually secreted in the stomach, while for trypsin, it is secreted by the pancreas. Pancreatic trypsin, I think. I think. You are making me do this. You're making me do this, huh? You're making me do this. Hold on. Trypsin. Because I cannot just give out false information. Bro, they didn't even say over here. You just said trypsin digests um, proteins into polypeptides. But sure. I think they're secreted by the pancreas. And ouch, okay. And the inactive form for pepsin is pepsinogen. And what activates it is hydrochloric acid. Optimum pH 1 to 2.5. For trypsin, inactive form is trypsinogen. It is activated by enterokinase, and its pH, optimum pH, is around 7, 6. No. 7 to 8.5, I guess, around there. Yeah. So that's the main difference between them. And lastly, we have peptidase. Converts polypeptides into digest polypeptides into proteins. Into amino acids. Sorry. Ooh. Okay. Lastly, we have our lipases. Oh no. I have to blow my nose. Lipases. So an example of lipases is stepsin, which is secreted by I think intestinal juices. Yeah, and they are responsible for the digestion of lipids into fatty acids and glycerol. Lipids. Yeah, yeah, intestinal lipase. There's no such thing as pancreatic lipid. Yeah, there's no such thing as pancreatic. I don't think so. So yeah, secreted by these small intestines. Okay, so you're done with the first. We took almost like 18 minutes just to explain the very first part of enzymes. Now there's two more parts of enzymes. I think we can brush through them pretty quickly. So the second part is how the enzymes work. So there are a few terms I need you to understand. First one is substrates. So substrates is basically substances that enzymes work on. They're called substrates. That's substrates. And secondly, we've got oh, what have we got? Oh, okay. So, secondly, we have to understand what's an active site. So, before that, substrates actually binds to the active site of an enzyme. An active site is essentially a like dentures. No, depression. Depressions on the surface of and oh my goodness. What is going on, huh? Oh my goodness. Okay, so active sites are basically depressions on the surface of an enzyme. The surface of an enzyme. So you see an enzyme, got depressions on it? Yeah. That's that right there. That right there is an active site, is the active site. Okay, so this part is a bit rocky, because when I was doing, I had to do quite multiple times just to grasp the concept. So enzymes are highly specific in nature, and what this means is that they're highly specific in nature. Highly specific in nature. So chemical reactions, um, uh, chemical reactions. What? Chemical reactions blank. Oh, think. Oh my goodness. Oh my gosh. Oh, you're down. Bye bye. Bye bye. Bye bye. Bye bye. And sorry, I didn't know I ended the video. Okay, sorry. Okay. Hi. My mic is still on. Okay. So what happens? What happens? What just happened? Okay, let's continue. Enzymes are highly specific in nature. Chemical reactions requires uh, chemical reactions of the substrate requires a unique enzyme, and the specificity of an enzyme is due to its specific three-dimensional shape. And what this means is that the substrate molecule must possess must have the or rather a complementary shape, uh, a complementary three-dimensional shape, a complementary shape to the active site of the enzyme, so that it can bind to it. That's what I'm going to next. Okay. So an enzyme, or rather, a substrate binds to when a substrate binds. I'm so sorry. Forgive me. I'm just saying it off the top of my head. This is not edited, by the way, so you're gonna have to watch me out, suffer through with me, okay? So, and a substrate, essentially, when a substrate binds with with an enzyme, enzyme substrate complex is formed. So when these two collide with each other and they collide properly and they actually hit the active site and it just nice fit, then an enzyme substrate complex is formed. And what happens is that the enzyme uh oh yeah, wait, okay, now is when I this is the part where I'm talking about the lock and key hypothesis. I'm so sorry. Okay, let me just sit down. The lock and key hypothesis. Bam, bam, bam. This one is a very important in an O-level biology examination. They ask you a question: lock and key hypothesis. Explain, describe. You must explain and describe properly using this a few steps. 所以哦，你要知道 and enzymes, okay, according to lock and key hypothesis, the lock, in quotes, is the enzyme, and the key is the substrate. Lock, enzyme, key, substrate. Remember that. Lock, enzyme, key, substrate. Okay. So what happens is that like what I said just now, when a substrate binds with the active site of an enzyme substrate complex is formed. Now the yeah, so basically fits lah. You see, like I'm just explaining. You don't have to say this in examinations. I don't believe you have to say this in examinations, but like I just want to explain that, like a lock, you're fitting into sorry, like a key, you're fitting into the lock, right? So the substrate, the key fits into the lock, which is the enzyme. Okay, I have to get it out because I'm still thinking. Okay, so this triggers the chemical reaction to start, and what happens is that during the chemical reaction, okay, this is not exactly what they say in the textbook, because in the revision guide, because I kind of forgot, but from what I can remember, it is yeah. So the during the chemical reaction, the enzyme converts the substrates into products. Okay, so that they can add that one line. I can check, actually. Hold on, because I don't actually believe in myself. I'm so sorry, guys. Oh my goodness, I'm a disappointment. Chemical reaction occurs at the enzyme's active site to convert the substrate into products. Chemical reactions occur at the enzyme's active site. And it converts the substrate molecules into products. Okay. After the reaction occurred, and the reaction is completed, the substrate molecules, the products leave the active side of the enzyme, and the enzyme is chemically unchanged. And okay, forgive me, I have to blow my nose real quick. Give me just 1 second. I'll just pause this real quick. Okay, I'm back. I came, sorry, I came from the toilet and I blew quite a lot of mucus out of my goddamn nose. Like, what is going on? What's going on? I'm not sick. I don't believe I'm sick. I need to take Zyrtec. Okay, where was I? I think it was after the. Okay, yeah, we're talking about after the reaction occurred. So the enzyme remains chemically unchanged and is free to catalyze other reactions. And what happens? What happens? What happens? What happens? It's free to catalyze other reactions. Okay. Okay. So the same enzyme can be used repeatedly to catalyze the same reaction essentially. And what this means is that a small amount of enzymes are needed to catalyze a large amount of um, substrates. To catalyze the reaction of a large amount of substrates. Okay, so catalyze the reaction. Let's put it out there. Catalyze the reaction of whatever. So at least, yeah, then it wouldn't be too awkward when you're writing it down. Okay, we're done with how enzymes work. I think so. Now it is time for the last component, which is factors affecting the rate of enzyme catalyzed reactions. Is it catalyzed or catalyst? Catalyze. Enzyme catalyzed reactions. Okay. Okay. So to put it out there, enzymes are actually temperature and pH sensitive. Wow. Okay. And they actually have an optimum temperature, an optimum pH in which they perform best in. So just now what I was saying was optimum, whatever, right? When I was talking about pH, optimum pH value, optimum pH level. I prefer to say level. I don't know. The book says value, but value dollar. It's a store. Okay. So oh my gosh, I cannot think. Let's talk about the enzyme's pH sensitivity. Temperature sensitivity. At low temperatures, the substrate molecules as well as the enzyme have low amounts of kinetic energy, and the the molecules, the substrate molecules as well as the enzyme, they basically move slower, and because of that, the chances of the molecules colliding with each other essentially is low, and hence the rate of reaction is lower lah. Now, as the temperature increases from low to optimum temperature, right? Low to optimum temperature, okay. Um, the the molecules will actually have um, an increasing amount of kinetic energy. The kinetic energy will basically increase, and this means that there's an increased chance of there's an chance increase in chance of the substrate molecules actually colliding with the enzyme and binding, or rather fitting into the active site of the enzyme. In the revision guide, I think they said something about like colliding with the active site of the enzyme and fitting into the active site of the enzyme. I don't know why they said it twice, but sure. If that's what you want to go by the book, then sure. Uh, now, this will mean that there is a large amount of effective collisions. Okay. Okay. Yeah. So, thus, the rate of reaction uh, increases. Now, they said something very interesting over here at this point. They said that for every 10 degrees 10, 10 degrees Celsius lah. For every 10 degrees Celsius increase in temperature, the rate of reaction of the rate of enzyme catalyzed reactions doubles. So this is kind of interesting, but it only applies to specific enzymes. We're talking about human biological enzymes. In this book, I think they're referring to enzymes in our body lah. So yeah, not really applicable to everything. I even searched in ChatGPT, and they said that it's not always like they always double like that when for every 10 degrees Celsius increase. But for O-level biology, I think it's safe to say that if you want to, you know, hey, I revised this and I noticed this. You know what? I'm smart. Yeah, that's what you want to. Yeah, so go by the revision guide, guys. This guide is reliable because it's from Dr. Ismail Hanif. Yeah, postdoctoral Harvard PhD BSc. Whoa, he got PhD. All right. He said, okay, he said, okay, he said, he said, he said, I say it in the exam, you get marks. Now, last point, in the human body, enzymes in the human body have an optimal temperature of 37 degrees Celsius. So that's why your body temperature is usually around that part. How do they maintain it even in cold weather? Well, homeostasis, which is a chapter in here as well. A whole lot of chapters, and I'm gonna talk about it someday. I don't want to think about it right now. Oh my gosh. Okay, so now we're done with increasing temperature from low to optimum temperature. Lastly, we've got uh, as the temperature increases past the optimum temperature, the rate of reaction actually rapidly decreases. So what happens at this point is that the graph from here, okay, is a zero, is like around here, right? It doesn't really reach zero yet, but it kind of like goes up and it all the way down until it reaches actual zero. So what happens is that high temperatures actually affect, or rather breaks the bonds that keep the enzyme in shape, that keeps the enzyme shape. Which, yeah, and what this means is that the substrate molecules can no longer bind into to the active site, because the active site basically loses its original shape. Active site loses its original shape. And because of this, yeah, the substrate molecules cannot bind anymore. Yeah, so therefore the rate of reaction basically decrease. By the way, the the enzyme is at this point, the enzyme is said to be denatured and can no longer catalyze the reaction, the chemical reaction of substrate molecules, and now they're kind of useless. Yeah. Mm. Uh, Enzymes are usually denatured at around like past above 60 degrees Celsius. Yeah, I just want to get it out there because it said in the book. So, by the way, now we're moving on to the definition of denaturation. Denaturation, I'm correct, right? Denaturation. I think so. So, denaturation is the change in the three. Okay, so denaturation is a change in the three-dimensional shape of the enzyme, um, as well as its active site lah. And oh no, I can't remember anymore. Yeah, okay, got it. And it is also it is caused. Okay, sorry. Yeah, so denaturation is the change of the enzyme's three-dimensional shape, and it is caused by the by heat or chemicals such as acids or alkalis. And yeah, okay, and alkalis. Okay, yeah. So this is about it. Yeah, and now we move on. We're done with pH sensitivity, temperature sensitivity. Moving on to pH sensitivity. I'm so sorry. it just came out of me. I'm so sorry. So pH sensitivity. As you know, enzymes are pH sensitive, and each enzyme has its own respective specialized or specific optimum pH level. So it's different. It may be different across many different types of enzymes. So, for instance, pepsin, its optimum pH is going to be 1 to 2.5, whereas for trypsin, it is going to be like around 6 to 7 to 8.5. I don't know if back then I said around, yeah, 7 to 8.5, yeah, pH 7 to 8.5. Whew. Okay, and uh okay, hold on. I'm thinking. I'll put this right. Okay. Okay. Yeah. Uh. Okay. Okay. I remember. Oh my gosh, I remember now. Okay. When the pH level deviates from the enzyme's optimum pH level, the rate of reaction actually rapidly decreases. So it kind of like goes like this. So for instance, if it's over here, the optimum temperature is over here, right? It kind of goes like this. So it's from zero up zero. So like, yeah, if you are to go, if you deviate from the optimum temperature, your reaction rapidly decreases. Now, extreme changes in the alkalinity or the acidity of a solution in which an enzyme acts on can actually cause the enzyme to be denatured as well. So if you have to change a particular solution's acidity, right, and after that, the enzyme in the solution, which is it like basically catalyzes the reaction in the solution, right, it's going to become denatured essentially. So it cannot catalyze the reaction in the solution. Yeah, that's about it. I think. Yeah. Yeah. Oh my goodness, what's going on? Yeah, let's go. I'm done. And how long did I take? I don't even want to know. I'm just gonna post this and upload it on YouTube and let the rest do the rest. All right. Peace out. This is for the chapter of enzyme, fully revised. I think all that's left is to do questions on it. But I think for now I'll leave the questions, the doing of the questions, the inventing of different questions in the revision guide. Because in the revision guide there is actually questions. But I'll leave that to later on at night when I cannot think, cannot memorize anymore. I'm just gonna write it down. Yeah. Okay. So I think I'll be doing. Yeah. Okay. So I think I'll stop with biology today. If there is time, I'll revise another chapter. But I think I should start revising my history. Not gonna lie. So outline of events next. Next event is going to be revision for. What chapter? It's going to be about the outbreak of war in Europe. There are like a few factors, and I kind of like started 1/3 of the way, and I was like revising the ineffectiveness of the League of Nations. Ineffectiveness of the League of Nations. I don't know why I said it so softly. Yeah. So that's about it right now. I'm dehydrated. I need water, and I'll come back telling you all about the outbreak of World War II and reasons for the outbreak of World War II in Europe.